Imagine looking at a map of a city. Right. But instead of just seeing the basic outlines of streets and buildings, you can suddenly zoom in and see inside every single house.
Right, like total surveillance.
Exactly. You can read the texts the residents are sending to each other, you can track exactly who's trading resources, and you can watch in real time as a coordinated crime is being plotted in a specific neighborhood.
It sounds like sci-fi.
It really does. But that level of omniscient, high-resolution surveillance is exactly what a technology called spatial omics has just brought to the human body. So welcome to the deep dive.
Glad to be here for this one. The transformation from traditional pathology to spatial omics it's fundamental.
Yeah.
Because for decades, analyzing tissue was basically like putting a sample in a blender.
A blender.
Yeah, literally. We could sequence a tumor, figure out all the mutations in that resulting biological smoothie, but we completely lost the geography of it.
Oh.
We had no idea if a specific immune cell was sitting right next to a cancer cell or if it was, trapped miles away behind a wall of dense tissue.
So the context was just completely gone.
Exactly.
Well, today we are diving deep into a massive 2026 review published in Briefings in Bioinformatics. It's by researchers Zerem, Fournier, and Selzit. And our mission today is to decode exactly how advanced artificial intelligence is being deployed to make sense of this sudden explosion of spatial data.
And it really is an explosion.
It is. And right away, the paper leans into this fascinating analogy. Okay, let's unpack this. They compare analyzing a modern human tissue sample to analyzing multispectral satellite imagery of Earth.
Which is a mathematically perfect parallel, actually.
How so?
Well, when a satellite captures geospatial remote sensing data, it doesn't just take a simple photograph. It records multiple spectral bands simultaneously to identify land use zones.
Like telling a city from a forest.
Right, distinguishing a dense urban grid from the surrounding trees. And the instruments we use now for spatial omics, they do the exact same thing to define what we call tissue domains.
We're talking about instruments like MERFISH, Codex, and MALDI-based imaging.
Right, exactly.
Because I was reading up on the mechanics here, and it's incredible. Codex, for example, uses this massive cocktail of antibodies that are tagged with unique DNA barcodes.
Yeah, it iteratively maps dozens of proteins.
And then MALDI uses mass spectrometry, which, and correct me if I'm wrong, literally involves firing a laser at a tissue matrix.
You're absolutely right. It fires a laser to vaporize the molecules.
That is wild.
It is. It measures their mass-to-charge ratio directly from the slide, which means we are mapping out these complex biological landscapes like the tumor core, the healthy stroma, the dead necrotic zones with just unbelievable fidelity.
But with all that detail, there has to be a catch, right?
Oh, definitely. The computational scale is what truly shatters traditional analytical boundaries. A single slide generates gigabytes of data.
Gigabytes from one slide.
Just one.
Yeah.
You're looking at hundreds of thousands of individual spatial locations. And at every single one of those coordinates, you're tracking tens of thousands of molecular variables simultaneously.
Okay, so that immediately explains why classical statistics just melt under the pressure here.
I absolutely melt.
You're dealing with what data scientists call the curse of dimensionality. Because if you try to run standard variance tests or linear regressions on 1 million pixels with 10,000 variables each, your computational matrix becomes overwhelmingly sparse.
Right, and highly nonlinear.
Yeah, the noise just drowns out the signal entirely.
Because classical statistics assumes a level of independence and normal distribution that simply does not exist in the chaotic, high-dimensional reality of human biology.
Biology is messy.
Extremely. So we are forced to deploy complex machine learning pipelines just to find the basic boundaries of these cellular neighborhoods.
So let's do a quick recap of where we are so far for you listening at home. We've got these high-tech laser and barcode instruments generating massive gigabyte-sized maps of human tissue. But the data is too huge for normal math to handle.
Exactly.
So we have to bring in AI. Let's unpack the baseline of that AI toolbox, starting with classical machine learning algorithms, things like k-means clustering and random forests.
Right, the foundational tools.
K-means is super common in data science for grouping similar data points. And I know it operates by iteratively calculating the mathematical distance between data points and central points called centroids.
Yep, that's the mechanism.
But the paper points out a massive biological flaw in how we historically applied it to tissue samples.
Yeah, because k-means relies heavily on Euclidean distance in its standard form.
Yeah.
So it inherently assumes that clusters are spherical.
Meaning it wants everything to be a perfect circle.
Exactly. It wants to group things into neat, equally sized biological bubbles. But tumors are aggressively irregular. They are not neat bubbles. Immune cells infiltrate in these jagged, chaotic lines. So when classical methods look only at the molecular profile and ignore the physical spatial coordinates, they produce what the authors call salt and pepper noise.
Salt and pepper noise. So you end up with a map where pixel segments are completely fragmented, like static on an old TV.
Precisely.
An algorithm might label a single pixel deep inside a healthy organ as a malignant tumor cell just because of a slight fluctuation in the local data.
Completely ignoring the fact that it's entirely surrounded by healthy tissue.
Which makes no sense biologically.
Not at all. So to solve that lack of spatial awareness, the field upgraded to deep learning, specifically relying heavily on convolutional neural networks, or CNNs.
Okay, here's where it gets really interesting. The paper focuses on architectures like UNet, CellPose, and Stardust. Let's talk about UNet because the mechanism there is fascinating.
It's a very elegant architecture. It uses a symmetrical encoder-decoder structure.
Right, the encoder path acts like a funnel. It aggressively compresses the image data to capture the broad global context, basically teaching the model like, okay, I am currently looking at a highly dense tumor region.
And then the decoder path symmetrically expands that data back out.
Oh, I see.
Yeah, it merges it with high-resolution features from earlier layers to draw incredibly precise pixel-level boundaries around individual cells.
It's like zooming out to see the forest and then zooming back in to draw a perfect outline around a single leaf.
That's a great way to put it. Pre-trained generalist models take this even further. They can segment densely packed cells even when their physical boundaries overlap.
Because classical thresholding, where you just set a brightness limit to separate a cell from the background, that fails completely when cells are squished together, right?
Completely fails. The cells just look like one big blob. But CNNs actually learn the morphological patterns. They learn the actual physical shapes of the cells so they can draw the borders accurately, even in a dense crowd.
But isolating a cell boundary is only step one. Because cells are highly social entities, right? They constantly secrete signals to influence their neighbors.
Oh, absolutely. Biology is all about communication.
So this is where graph neural networks, or GNNs, enter the pipeline. While a CNN finds the cells, a GNN maps the society.
I love that. Yes. GNNs execute a process called message passing.
How does that work?
Well, the network treats every single cell as a node. And the algorithm iteratively updates the state of a specific node by mathematically aggregating the feature vectors of all its immediate neighbors.
Wait, break that down for me.
Sure. It basically looks at a cell, then looks at all the cells touching it, reads their data, and uses that to update its understanding of the first cell. It learns not just what a cell is, but what its exact microenvironment looks like.
Got it. So it's a difference between analyzing an individual in complete isolation versus analyzing their social network. Classical machine learning is like sorting people by their zip code based on their income and age. It's useful, but it's static. But a graph neural network is like tracking who is actually talking to whom at a crowded party. It identifies the influencers, the isolated cliques, and the gossip networks spreading information across the room.
And the predictive power of mapping those biological conversations is immense. We can pinpoint exactly where an immune cell is receiving a suppressive signal from a tumor cell.
That's incredible.
It is. But mapping these networks introduces a severe structural bottleneck for the technology.
Which brings us to the data hunger of deep learning. These models require massive amounts of annotated data to learn what a tumor or a T cell actually looks like.
Yeah, they are ravenous for data.
But expert annotated medical labels require a highly trained pathologist to sit at a screen and manually draw circles around thousands of individual cells.
Which takes forever.
It's an unsustainable, super expensive bottleneck. So, just doing another quick 5-minute recap here. We moved from basic clustering that caused static to CNNs that perfectly draw cell borders, to GNNs that track how cells talk to each other.
Right.
But now we are stuck because the AI is starving for human-labeled training data.
Exactly. So to bypass that lack of human-labeled data, researchers deploy generative models.
Like what?
Variational autoencoders, or VAEs, and generative adversarial networks, or GANs. These architectures learn the underlying statistical distribution of the tissue data.
And they use them to fix problems in the data, right?
Yeah, they are heavily used right now to correct batch effects, Those are the technical artifacts and random variations that pop up simply because a tissue sample was processed on a different machine or maybe in a different lab.
Okay, I want to focus on GANs for a second because the mechanics are crucial to a major risk we really need to discuss. A GAN consists of two neural networks locked in a game, right?
Yes, a zero-sum game.
You have a generator acting as a counterfeiter, trying to synthesize fake biological data, and then you have a discriminator acting as a detective, trying to flag the fakes.
Right, and they compete until the generator produces synthetic tissue data so realistic that the detective discriminator is entirely fooled.
Okay, but wait, I really need to push back on this application based on a terrifying detail I read in the text.
Uh-oh.
The paper explicitly warns about something called hallucinated structures and mode collapse.
Ah, yes.
If we are using a counterfeiter algorithm to fill in the gaps of our medical data, are we risking the AI literally inventing a tumor structure or a biological pattern that simply does not exist?
You've hit on it. The danger of biological hallucinations is easily one of the most critical vulnerabilities in the entire field right now.
Explain mode collapse to me.
Mode collapse happens when the generator realizes that producing one specific, highly convincing fake pattern constantly fools the discriminator. So it just stops exploring the full complexity of the biology.
It gets lazy.
Exactly. Finds a cheat code and spams it. If the architectural priors of the model fail to capture the messy reality of human tissue, it will confidently output synthesized data that is biologically implausible.
So imagine an AI generating synthetic training data that accidentally hallucinates a novel type of blood vessel.
Right.
And then an automated diagnostic tool trains on that hallucinated blood vessel. If a clinician subsequently bases a chemotherapy dosage on that model's output, the consequences are catastrophic.
It could cost lives. And this interpretability crisis is exactly why the review emphasizes the absolute necessity of explainable AI. We cannot integrate black box neural networks into clinical oncology.
We have to know why the AI made the decision.
Exactly. The authors highlight algorithms like Lyme, saliency maps, and particularly SHA values.
Yeah, SHA values are brilliant. The acronym stands for Shapley Additive Explanations, and it actually borrows directly from cooperative game theory.
It does.
It basically treats the AI's final prediction as a payout, and then it calculates exactly how much each individual pixel or molecular variable contributed to that final payout.
It forces the AI to show its work, basically, to highlight the exact features that triggered its decision.
And the paper also mentions model distillation as a safety mechanism. How does that work?
Model distillation is really cool. You take the massive, unreadable deep learning model, we call it the teacher, and you use it to train a smaller, simpler surrogate model.
The student.
Right, the student. And this student model relies on human-readable decision trees. So it maintains high accuracy, but it actually allows a clinician to audit the logic path.
Okay, so if we have this massive toolbox from classical clustering to hallucination-prone generative models to game theory explainers, a bioinformatician or a clinical data scientist is left with a pretty daunting choice.
Very daunting.
How do you know which tool to deploy without over-engineering the solution? Well, the authors provide a brilliant, highly structured decision framework in the paper. It's built on a 5-axis matrix.
Modality, resolution, tissue organization, annotation availability, and clinical objective.
Let's walk through the mechanics of some real-world scenarios they lay out so you listening can actually apply this.
Sure, let's start with scenario A. Let's say you are working with low spatial resolution data and a single modality. For instance, you're only sequencing RNA and the data is relatively scarce.
The framework dictates that you stick to classical machine learning there. You use k-means or random forests.
Exactly.
Because throwing a deep convolutional neural network at low resolution, single modality data is computationally wasteful and practically dangerous because a deep learning model will likely overfit the noise, creating patterns out of pure static.
Now consider the exact opposite end of the spectrum, scenario B. You have multi-omics data.
Multi-omics, layering multiple types of data.
Right. You are layering transcriptomics, proteomics, and metabolomics. You are looking at the genetic code, the physical protein structures, and the metabolic exhaust of the cells all at the exact same time.
For multi-omics, the framework points straight to cross-modal transformers or optimal transport alignment.
Optimal transport is fascinating.
Let's ground that with a medical example. Imagine mapping a dense pancreatic tumor. You need to see the physical protein wall that the tumor built to block chemotherapy, right? And simultaneously, you need to map the genetic mutations driving the cells inside that wall. Optimal transport is a mathematical technique, often compared to the Earth mover's distance.
Which is such a great visual.
It really is. It calculates the absolute most efficient way to morph one probability distribution into another. So it allows an algorithm to perfectly align the protein map onto the genetic map without warping the underlying spatial topology.
It creates a perfectly synchronized multidimensional map. It's beautiful math. But scenario C introduces an entirely different barrier: clinical translation across institutions.
The privacy issue.
Exactly. Your objective is building a universal diagnostic model, but your data is siloed. You have patient tissue samples in a hospital in Paris, a research center in New York, and a clinic in Tokyo.
And strict privacy laws like KPay in the US or GDPR in Europe mean you absolutely cannot centralize gigabytes of raw patient genomes onto a single server.
You'd go to jail.
Yeah. So the decision matrix dictates you must deploy federated learning.
Federated learning decentralizes the training process. The raw patient data never leaves the local hospital's secure servers. Instead, the global AI model itself travels to the hospital.
Oh, the model travels, not the data.
Exactly.
Oh.
It trains on the local data, updates its internal mathematical weights, and then sends only those updated weights back to the central server that gets aggregated with the learnings from other hospitals.
To bring back our earlier social network analogy, federated learning is like trying to figure out the general vibe of several different crowded parties across the city.
I like where this is going.
Instead of forcing the host to give you their private guest lists and text transcripts, you just send an observer to each party. The observers report back and say, hey, the trend tonight is jazz music and dancing, completely preserving the anonymity of the actual attendees.
That's exactly it. It allows the medical community to build globally intelligent, highly robust foundation models without compromising patient privacy.
And there's always but even with these optimized frameworks, we are rapidly hitting the physical limits of our current hardware, aren't we?
Oh, we're slamming into a wall.
Yeah.
The sheer density of organ-scale multi-omics is literally melting standard GPU clusters.
So to recap where we are now, we learned how to pick the right AI tool based on the data, from basic algorithms to multi-omic optimal transport to privacy-safe federated learning. But now our computers are too slow.
They just can't handle the math anymore.
Which takes us to the horizon of the field, the sci-fi stuff. The paper outlines some wildly futuristic cross-disciplinary leaps designed to bypass those silicon limits, starting with quantum computing.
Yeah, the application here is quantum-inspired machine learning and quantum support vector machines.
Break that down for us.
Classical computers process information sequentially, evaluating one state at a time. But quantum algorithms can leverage superposition.
Being in multiple states at once.
Right. They use superposition to map these high-dimensional spatial omics data sets into theoretically infinite-dimensional latent spaces, so they can evaluate massive cellular atlases simultaneously. While purely quantum hardware is still maturing, researchers are actually designing hybrid workflows right now to offload the most mathematically paralyzing matrix inversions to quantum circuits.
That is mind-bending. But the cross-disciplinary borrowing doesn't stop at quantum physics. I was fascinated by the adoption of a concept called desintering.
From materials science, right?
Pulled straight from advanced manufacturing. In manufacturing, sintering is the process of applying heat to metal powders until they fuse into a solid mass. Desintering is the mathematical modeling used to virtually separate those fused parts.
And the biological translation of that concept is incredibly clever.
How are they using it?
Well, when researchers grow dense tumor organoids in a lab, the cells replicate so aggressively that under a microscope, their nuclei appear completely fused together.
Like the metal powder?
Exactly like the metal. Classical thresholding algorithms treat the entire mass as one giant deformed cell. But by applying morphological opening and closing operations derived from materials science desintering.
Algorithms can computationally unstick the overlapping nuclei.
Yes, allowing us to profile the cells individually.
It's a perfect example of how bioinformatics is just absorbing every scientific discipline available.
Truly.
But perhaps the most profound frontier discussed in the review is the integration of game theory. We touched on this earlier with the SHEP values, but the implications are staggering here. We are fundamentally shifting from viewing a tissue sample as a static snapshot to modeling it as a dynamic, ruthless economy.
Because cells are not passive structural building blocks. They are independent agents playing a continuous game of survival.
It's basically a war zone in there.
It is. Game-theoretic models analyze tissues by calculating the specific strategies cells deploy, like tumor cells competing aggressively for limited oxygen and nutrients, or cooperating in complex signaling networks to actively suppress and evade infiltrating immune cells.
And to simulate this biological game, researchers are building massive foundation models using self-supervised learning, or SSL.
SSL is the game changer.
Instead of relying on humans to painstakingly label thousands of images, SSL feeds millions of unannotated spatial omics maps into an algorithm and forces the model to mask and predict parts of its own data.
Right, like blanking out a word in a sentence and guessing what it is.
Exactly. And through that sheer volume of self-prediction, the model organically learns the universal rules of the game. It learns the underlying grammar of how human tissues organize, how cell neighborhoods form, and how diseases structurally manipulate their environment.
The convergence of scalable compute, multi-omic spatial profiling, and self-supervised foundation models is rapidly pushing us past mere observation. We are moving toward predictive simulation.
Which will redefine precision oncology.
Over the next decade, absolutely.
Which leads to a massive lingering implication for you to consider as we wrap up this deep dive. If we view tissues as dynamic ecosystems playing out a competitive game, and our foundation models are rapidly learning the universal rules of that game, how far are we from executing a full digital twin simulation of a patient's exact tumor?
That as far as you might think.
Imagine a clinician mapping your biopsy, loading that high-dimensional spatial data into a simulation, and virtually testing a dozen different chemotherapy combinations.
Running simulations before touching the patient.
They could watch exactly how your specific tumor's cellular network adapts, mutates, and attempts to evade the treatment, allowing the doctor to adjust the strategy and literally win the game before a single drop of real medicine enters your body.
From a static satellite image to a predictive digital simulation of life itself.
Something to think about the next time you look at a medical scan. Thanks for joining us on this deep dive.